Hello, and thank you for joining me for the third installation of the Luxendo Lightsheet webinar series. My name is Dane Maxfield, and I'm a Sales and Applications Specialist with Bruker. Today, I'm going to be speaking to you about cleared tissue imaging with lightsheet fluorescence microscopy. Before I get going, I want to point out the questions window in the bottom. If you have any questions during the presentation, please feel free to type them in there, and I'll do my best to address them either during the presentation or at the end. If it pertains to a particular slide, please reference the slide number for me. For today's presentation, here's a brief outline of what we're going to cover. First, I want to give you an introduction to lightsheet microscopy, just kind of remind people the benefits of this technique and why we like using this for our 3D imaging. Then I want to touch on optical clearing, why we do this, some of the techniques using it. It's going to be a brief overview, but it will give you an idea of some of the advantages of this. And then we'll touch on the challenges of imaging these cleared samples, things like refractive index, working distance, and sample size. For the back half of the talk, I'm going to introduce you to our new lightsheet system, the MuVi SPIM for cleared samples, or MuVi SPIM CS, and talk about some of the ways that we work with refractive index, sample size, sample mounting, and then go over the easily exchangeable lenses so you can flop between cleared tissue imaging and live cell imaging. And last but not least, we'll go through some applications data showing you some of the images that have been acquired by our early customers and collaborators. So to begin, let's remind everyone what lightsheet microscopy is. The basic premise and principle of a lightsheet system is that we're going to take your standard microscope imaging pathway and we're going to decouple it into separate fluorescent excitation and fluorescent detection pathways. And in doing so, we'll orient these orthogonally to each other such that the excitation pathway comes in and specifically illuminates the focal plane of interest of the detection objective. And this gives rise to the acronym selective plane illumination microscopy. And people will use this interchangeably with lightsheet. So although this technique is, I guess, not quite a century old, but the root of it is over a century old, the lightsheet really didn't hit mainstream until the early 2000s. When Jan Huisken and Ernst Stelzer published their landmark paper introducing lightsheet fluorescence microscopy as a technique for fast and gentle 3D imaging. And why people want to use lightsheet, in order to understand this, we really need to understand the drawbacks of conventional imaging. Say, for example, we want to image the focal plane highlighted in red of this cartoon Drosophila embryo. With a laser scanning confocal, what we'll do is we'll bring in an excitation beam, either from above or below, that will travel through the embryo to the focal point, and then we will raster scan this point by point across the embryo and build our image in a serial fashion, again, point by point. In doing so, what we end up doing is exposing the entire sample to light, even though we only let a subset of that light to our detector as determined by the confocal aperture downstream. Spinning disk, we get a little bit of speed from this by parallelizing the excitation using multiple beams from a disk of pinholes and parallelizing the detection by using a camera, but the same drawback applies. We excite the entire embryo while only letting a certain subset of that make it to our detector as determined by the disk of pinholes. Conversely, in lightsheet, when we want to excite the focal plane in red, we'll bring in our excitation light orthogonally to how we detect, selectively illuminate that focal plane of interest, and then capture that optical section in one shot using a camera. This gives us two major advantages compared to the confocal techniques. First of all, speed. So if we're trying to image the embryo, the total imaging time for a light sheet is significantly faster than a laser scanner or a spinning disk. This is in part due to the illumination scheme. Because we have essentially a wide field based illumination, that is, there's no confocal aperture downstream, we can use a much lower laser light and detect with much higher signal to noise than we can in laser scanner or spinning disk. The second advantage is the phototoxicity and photobleaching. Because we're only selectively illuminating the focal plane of interest and not anything outside of the focal plane of interest, we are effectively using our fluorophores to the best of their ability. We are not exciting any fluorophores we're not detecting, and we're not going to be exposing the embryo to more light than it needs, inducing phototoxicity. So again, some of these key advantages just written out here is dramatically reduced light dose, which means less photobleaching and less phototoxicity. We get the detection of the optical section in a single shot, which means much higher imaging speed, and we can do this without sacrificing 3D optical resolution. That is, we get the same advantages of a confocal with the speed and with less photobleaching and phototoxicity, making this ideal for rapid imaging of large non-scattering samples. Examples of this include Drosophila embryos, zebrafish embryos, and given the title of today's presentation, also cleared tissue. But the key phrase in this is non-scattering. However, most biological tissue is scattering. And what that means for your imaging, if we consider the cartoon mouse embryo in the upper right corner, is if we're trying to image this with a microscope, we see that light has a very difficult time penetrating into the tissue. Stereotypically, visible light can travel 150 microns to 200 microns into a tissue, again, depending on the opacity, before it's scattered. And when it comes to imaging, this scattering has a twofold effect. Not only do we have difficulty getting photons into the sample to excite the fluorophores, but we also have difficulty getting photons out of the sample and collecting them on the detector. And this is where optical clearing comes into play. By clearing the sample and matching the optical properties with the microscope objective and the media, we can now image many millimeters through these cleared samples, allowing tissues, organs, and even organisms to be imaged with 3D microscopy techniques. Now, as you might imagine, making something optically clear is not a trivial process. This typically involves chemical treatment of the tissue, and specifically fixed tissue, to make the sample optically transparent while keeping the structure intact. And that last part is important. I'm sure there's many ways to make something optically clear, but just turn it into a primordial soup. And we want to be able to keep that structure there in order to get that information. Otherwise, there's easier ways of acquiring it. The second thing is we want these clearing techniques to be compatible with fluorescent labeling strategies so that we can look inside the specimen and then image these with 3D microscopy. Optical clearing in a nutshell, and again, this is a gross oversimplification of this. There are many detailed reviews out there that will go into these protocols in much more detail. But there's typically two steps. So if we take our opaque mouse brain here, the very first step that you'll do for an optical clearing is to do some sort of lipid removal. So we'll incubate the sample in organic solvents or detergents like SDS in order to remove the lipids and all of these scattering tissue or scattering elements and get them out of the sample. The second step is typically a sort of refractive index matching. So, and I'm going to go into this in a lot more detail in an upcoming slide, but the idea is that we want to match the tissue and the media that it's in order to create an optically clear brain that we can image through. Now, as you might imagine, given the vast number of samples, there are also a plethora of different optical clearing methods out there. As a rough pass, we can group these into 4 or 5 different groups. Simple immersion methods such as CDB and fast clear, which are essentially refractive index matching. We're not doing too much lipid removal there. There's organic solvent-based methods like microdisco, 3-disco, and BAB, which use these harsh organic solvents to remove the lipids and a refractive index matching afterwards. Hyperhydration, which uses a little bit gentler solvents like SDS and refractive index matching afterwards. And then there's other techniques that use hydrogel embedding, so making a tissue gel hybrid or matrix in order to hold the tissue in place while we move away those scattering elements. And again, many others that aren't even covered here. The thing I want to highlight for people is that no single clearing method will work for all tissue types, sizes, or experiments. And this is where a week in the library can literally save you a year in the lab. You got to do your literature searches, see what works for your samples. There's multiple reviews out there, so I'm sure that there's something close to what there is, and you can adapt those protocols based on your particular sample. But even when we have a clearing method that works well for our particular sample, have it cleared and are ready to image, there are a couple key things regarding the microscope that we have to keep in mind. First is sample mounting. So now that we have these cleared samples, which are typically large and mechanically delicate, we need to be able to put them onto the microscope and reproducibly position them in order to do 3D imaging. So we need a reliable, stable mounting strategy for these cleared samples. Secondly, and I think this is one of the most overlooked things in microscopy, is the refractive index. As a reminder, changes in refractive index can cause bends in light. So if we see our light ray coming in through a low refractive index media here, as it hits a barrier with a higher refractive index, we'll see that theta 1 and theta 2 no longer match, so the input angle and the output angle, showing that this light was actually bent once it hit that barrier between the refractive indices. What this means for your imaging is that you can actually, your objective will actually be seeing different focal points depending on where the rays are coming. So these low NA rays coming from the center of the objective come to a focus point that's farther away than the high NA rays that will focus closer. So this can cause low order aberrations and a significant degradation of image quality. Because of this, there's two things we need to think about with regards to refractive index. We need to be able to match the refractive index of the sample and the imaging media together in order to avoid these. And secondly, we need to be, the refractive index of the sample and the media must also match the workable range of the objective that we're using for the collection of these images. And with large samples, we're going to need long working distance objectives. So these are typically air or water multi-immersion objectives. And the big thing with these is making sure that we have enough working distance so that our sample doesn't bump the lens and we can image through it. With all of these challenges in mind, we've now developed an extension off of our current multi-view SPIM and have the new MuVi SPIM CS, or MuVi SPIM for cleared samples. And what this is a new design as far as optics, working distance, sample size, and sample mounting strategies specifically geared around imaging cleared samples. What stayed the same between the MuVi SPIM and the MuVi SPIM CS is the excitation pathway. So we're still going to be creating our light sheet with digitally scanned Gaussian beams, and we'll have a variable thickness of this beam that's adjustable based on user preference, anywhere from 2 microns to 8 microns. The excitation is going to come from 2 separate illumination arms consisting of 2 10x lenses. And the reason we do this is to extend the depth of field for our light penetration for excitation. And if we create light sheets from 2 objectives on either side of the sample, we can integrate these and create a longer, more even sheet over a greater distance and get better light penetration into the sample. What we did change on this is the detection optics. So now we're working with 2 lenses to begin with. We have a Nikon 20x 1.0 NA multi-immersion lens and a Nikon 10x 0.5 NA multi-immersion lens. With the magnification changer that's on the MuVi, what we can see is we can choose between the type of optical resolution we want, 300 nanometers for the 20x or 600 nanometers for the 10x, and then choose the field of view and the sampling by changing the magnification downstream without actually changing the lens. These lenses also have a very long working distance, 8 millimeters for the 20x and 5.5 millimeters for the 10x. And they work with a pretty wide range of refractive indices, 1.44 to 150 for the 20x, and a really big range, 133 to 151 for the 10x. And what we can do with these lenses is each one of them has a correction collar, which allows us to correct for these changes in refractive index in order to focus these rays so that they are both hitting the same position or collecting light from the same position inside the sample by adjusting that correction collar. So that's one of the ways that we go about optimizing this for refractive index. And last but not least, we've got a couple different detection optics schemes for built around your particular sample. In this, we can have a single-sided chamber or a dual-sided chamber. The dual-sided chamber, consisting of 2 10x's or 2 20x's, or even an unbalanced configuration with 1 10x and 1 20x, will have accommodate samples up to 12 millimeters between the illumination lenses and 8 millimeters between the excitation lenses, while being 19 millimeters deep. If that's going to be a little restrictive for your sample, we have 2 other octagon options as far as lens configurations that will extend the width of that chamber up to 16 millimeters with the 20x lens. And even with the single-sided octagon, we can still image both sides of the sample by imaging one half, rotating it 180 degrees, and then imaging the other half. And with our GPU-enabled data fusion pipeline, we can then take these 2 image sets and create a single high-resolution data set. For sample mounting, there's a couple different options. Here I'm showing you one of our platform options in which we'll actually just take the cleared tissue and place it on a platform. We can then lower this between the objectives and do our imaging. Some other options for mounting include a hook, skewering the sample, hanging it by supergluing it to a disposable element, or even a cuvette. And the nice thing about this is, since the MuVi was originally designed for live cell imaging, we can change octagons to go between live cell imaging and cleared tissue imaging in a matter of minutes. And to highlight this, I'm showing my shiny-headed colleague here, Jürgen, who's going through and is changing the octagon from a live cell imaging set of lenses to a cleared tissue imaging set of lenses. And from starting this, changing the lenses, aligning them, and getting the beams co-aligned, it takes him a little bit more than 10 minutes. So pretty easy to exchange these different lenses. And now for the fun part, let's show you some of the data that we've collected with our early customers and collaborators. We really feel that this system is going to be make a mark in neuroscience, developmental biology, cancer biology, and some pharmacology and medical research. And this is because these fields typically require imaging of these large samples and and some 3D mapping of the sample. So to start with, we've got octopus up here. It's been labeled with a red membrane marker, and this was taken with our 10x lens. However, if you have samples that don't fit in the entire field of view, we have a tiling and stitching platform or add-on for this that allows you to take images from multiple different points and stitch them together to create these huge panoramic fields of view. To highlight this, we have a mouse head here. Here we've labeled the developing neurons in green and the differentiating muscles when bred. And this is a 6x5 tiled image, highlighting the ability to capture these really large fields of view at very high resolution. Here I'm showing you a cubic cleared brain. So we're imaging about 10 millimeters deep into this. We're seeing the axons and dendrites here labeled with GFP, and you see a very high density of these axons. And if we focus on a little bit lower density area, we'll see that we can image and actually trace these different neurons. And here what I'm showing you is a maximum projection image that's color coded based on depth. So the warmer colors are towards the top of the image and the cooler colors towards the bottom. And this will give you an idea of some of the 3D information that's still contained in this single max projected stack. And finally, we've got a mouse lymph node here that we've reconstructed in 3D and are just rotating around so that you can get a feel for the large types of images that we can reconstruct with this. And to remind you, this is the 4th addition to our Lightsheet platform. So we currently have the MuVi SPIM and now the clear tissue extension of this, the MuVi SPIM CS. We also have our inverted SPIM, which is great for monolayers and mouse embryos, and our upright geometry, the QV SPIM. So take-home messages from today's presentation. First, I hope that I showed you that Lightsheet microscopy is suitable for your day-to-day imaging of both live specimens and cleared samples. Secondly, I've introduced our new MuVi SPIM cleared sample, which provides solutions for imaging these large tissues and changes the way we do our sample mounting and the optics involved in this. And I hope I've shown you that the very fast, easy octagon exchange makes for a quick transition between live cell imaging and cleared tissue imaging that will really be a benefit for people that are trying to do both. And with that, I'd like to thank everyone for their attendance, and I will take any questions. Thank you. Okay, so we have one here. What about imaging in organic solvents with a higher refractive index of about 1.56? So that will require a different lens set. So we will have a cuvette solution for that. The organic solvents that typically aren't, the lenses that we're using, aren't really good for imaging in these harsh organic solvents, and so we'll end up keeping that sample in a cuvette and using different lenses to image with that. How do we fix live imaging? How can we do live imaging if we have to fix tissues? That's the conundrum here. So the idea behind this light sheet is that it will switch between live cell imaging and cleared tissue imaging, but those are typically mutually exclusive applications. Biggest sample we can put in the chamber, that is a 12 millimeters wide and 16 millimeters long and 19 millimeters deep. And so that will, and that is the single-sided imaging chamber. How's the resolution compared to confocal microscopy? That's a good one. So with these, the resolution is based on the numerical aperture of the lenses that we're using. In this particular case, the highest NA lens that we have for imaging these cleared tissues that have long working distance is a 1.0 NA lens, and so that's going to give us approximately 300 nanometer spatial resolution. Our axial resolution is determined by the light sheet thickness, so it will be approximately 2 microns. Do maximum magnification and resolution. Maximum magnification at the moment is 30x with a 1.5 mag changer on there, and then we will change that to, or that will be upgradable to a 40x total mag as well. With a single-sided illumination, can the light penetrate all the way through if the sample is cleared? Again, it depends on the sample. So the dual-sided illumination just gives you the benefit of being able to excite from both sides, and that gives us a little bit better light penetration. So if there is something that's scattering or absorbing on one side, we might be able to make it through on the other side with the second illumination beam. How many excitation colors are available? There are up to 6 laser lines that you could put in. And if there are many, how do you deal with chromatic aberrations and the light sheet alignment? The light sheet alignment is, for the most part, mechanical. We are working on solutions to deal with how that changes, how the beams change with the refractive indices. And the chromatic aberrations, that's mainly a function of the objective. So the objective that we use is highly corrected for visible light, but it's not very good for UV. It's the max sample size you can have for a double-sided illumination. I think you mean double-sided detection, and that would be 8 millimeters wide between, or on the small axis is the max sample size. For big samples, how do you reduce shadowing and stripes? The way that we go about doing this is rotating the sample slightly. So we can rotate and take this from multiple views and then fuse this together to create a high resolution image. And I am not seeing any more questions come in. If there are any, my email is down there, or you can feel free to contact your local Bruker rep and they will direct you to the appropriate person. Thank you very much.